today we had completed the transition. If you look into the proportions we have completed that how DNA replication can be restricted, loading can be sorry. How, just a second, let me get my pointer. The loading of DNA is restricted to early G1 and the initiation restricted to S. And we also studied how the cell passes through the restriction point. We studied the role of retinoblastoma and we studied the role of cyclin D. So we have done this transition. We have understood the replication of DNA being restricted, loading being restricted to early G1 and initiation to S. And now we'll go to another vital transition that is operative in cell cycle, is the passage of the cell from G2 via M to G1. Now, when we are talking about the passage of the cell from G2 via M to G1, there are three notable experiments that came up with this important fact that there is a key regulator, that key molecules that is responsible for the control of mitosis. I'm not going to go over the three experiments, but this is one of the experiments that was done in 1971, which showed that in mitosis, some key molecules play a very important role in controlling the passage of the cell from G to MG1. Now, what was the experiment? This experiment, as I said, was done in 1971 by two independent research teams led by Yoshui Masui and Clement Market, as well as Dennis Smith and Robert Ecker. What they did is that when we started cell cycle, I had said that oocytes are arrested at G2. When the hormone progesterone is available, the oocytes mature and then they enter the M phase of meiosis. So naturally, the hormone progesterone actually triggers via which the arrested oocytes can mature and enter into the M phase of meiosis. This is natural. So what was done is that in this experiment, the frog oocytes which were arrested in G2 were taken and they were administered this hormone progesterone. And in presence of progesterone, what happened is that these arrested oocytes matured and entered into the meiosis. So it was evident that hormone progesterone triggered meiosis. Now the question was that whether this hormone progesterone alone is itself responsible for induction of meiosis or some other molecular player is simultaneously playing an important role. To study that, what they did was that a frog oocyte which has entered meiosis as a result of induction via progesterone, the cytoplasm of that oocyte was taken. And this cytoplasm was then injected into the cytoplasm of another oocyte which was similarly arrested at the G2 stage of cell cycle. When the cytoplasm of that cell that has entered meiosis was injected into the cell of the egg where the oocyte was arrested at G2, it was observed that such cytoplasmic transfer induced the G2 to M transition in absence of hormonal stimulation. So once more, in the first experiment, progesterone stimulates the transition from G2 to M. The cytoplasm of this cell is taken and injected into a oocyte arrested at G2. And then it was observed that automatically such cytoplasmic transfer resulted in induction of the arrested oocyte at G2 to M, which clearly indicated that progesterone is not the sole element that is actually inducing the arrested oocytes to enter M phase. There are some cytoplasmic determinants present in the cytoplasm, some cytoplasmic factor present in the cytoplasm, which plays an important role in the transition of the cell from G2 to M. Since the entry of oocytes into meiosis is frequently referred to as oocyte maturation, This cytoplasmic factor was then called as maturation promoting factor. I'm not getting into the details of the next two experiments which further elucidated the importance of MPF. Let us see what is actually this maturation promoting factor. So when this maturation promoting factor was studied in detail, again it was seen that it was a partner complex. The partner complex consisted of a dimer, which consisted of cyclin B, which was the regulatory subunit, and it consisted of the enzyme CDK1, which was the catalytic subunit. Just like we had studied the role of cyclin B CDK4/6 in G1S transition, where I had said that the cyclin B CDK4/6 are partners. Similarly, this maturation promoting factor is nothing but a partner protein consisting of cyclin B, which is regulatory and which can be easily degraded. It can be synthesized on the basis of certain environmental stimuli and can be very easily degraded. So this is comparatively a stable enzyme part or the catalytic part, which will bring about the phosphorylation of the substrates. Keep it in mind that CDK1, so it's cyclin dependent kinase. The name suggests that this enzyme's activity is dependent on the cyclin counterpart. Secondly, the name is cyclin dependent kinase. If you remember what we studied in cell signaling, kinase is such an enzyme that activates its substrate by adding phosphate to it. So this CDK1 is an enzyme that is dependent functionally on cyclin B. And how does it activate its substrate? It activates its substrate by adding phosphate to the substrate. Now, this is a very rough picture on the right side, showing you how the cyclin B is present during the cell cycle. You see, the moment the cell is coming out of mitosis, the mitotic Cdk. So if I consider to be the maturation promoting factor, then this will be the mitotic cyclin, and this is the mitotic Cdk. You see that the moment the cell is coming out of mitosis, the mitotic Cdk, in this case it is CDK1. The CDK1 is alone and the cyclin B part has degraded. So the cyclin has degraded, and as it travels through G1, S, G2, it is all alone. So as I said, it cannot function alone. It remains alone and it passes through G1, S and G2. The moment the cell enters G2, you can see that mitotic cyclins during this process has been synthesized, and this mitotic cyclins then bind to the mitotic Cdk just at the outset of mitosis, and this partner pair is activated and then the cell enters mitosis. So it is the presence of cyclin B and the absence of cyclin B that actually controls the CDK1 activity. When we have an active cyclin B CDK1 complex, we have to assume that the MPF is active, the cell is in mitosis. Otherwise, whenever there is this inactive CDK1, we have to assume that the cell is not in mitosis. It might be in G1, S or G2. So we understood the importance of this partner complex cyclin B CDK1. Now let us say at the molecular level, what is that event that actually activates this? Just a second. That actually activates this mitotic cyclin CDK partner complex, and at the outset of mitosis, and the cell enters mitosis, and then with the degradation of cyclin B, the cell exits mitosis. So there is a molecular event that is actually controlling this phenomena. So let's look into the molecular event, but before moving into the molecular event, this is just a recapitulation of what you have done. This is to recapitulate that what the enzymes we are studying now are kinases. So whenever there is a kinase protein acting, what happens? It adds phosphate to its substrate. In the process, it is activating its substrate. If an activated substrate needs to be deactivated, then the second protein, that is phosphatase, comes into play, which removes the phosphate. So there is a set of enzymes which are kinases, which adds phosphate to the substrate, thereby activating the substrate. Now, if we want to deactivate the substrate, we need to remove that phosphate. Now, the removal of the phosphate is done by protein phosphatase. This is something that we have learned in our signaling classes. So this is a recapitulation. And now look into the molecular mechanism. Let us concentrate from, let's start our story from here. Cyclin B is degraded. The moment cyclin B is degraded, as you can see, it is being degraded by a complex written here, APC/C. This is an anaphase promoting complex. We'll come to that later on. I'm not getting into that complex today. Now, the moment cyclin B is degraded, you have to understand that the cell has come out of mitosis. As I said, the moment the cell comes out of the mitosis, the CDK1 partner is all alone. Now concentrate on CDK and look at the 3 amino acids present in this position. 161 position, 14th position, and 15th position. Remember, cyclin dependent kinase is an enzyme. It is a polypeptide made up of a number of amino acids. There are 3 amino acids in 3 certain positions that plays a very vital role in controlling its activity. The 2 amino acids in position 14 and 15. In position 14, we have the amino acid threonine. In position 15, we have the amino acid tyrosine. And in position 161, we have the amino acid threonine. So it is the 3 amino acids phosphorylated or dephosphorylated state that plays an important role in controlling the activity of CDK1. So if we have understood that these 3 important amino acids are important, now let's again start the story. Cell comes out of mitosis, cyclin B is degraded. Now look at the state of CDK1. You can see that of the 3 amino acids I said important, threonine 161 is still in the phosphorylated state. What happens? In the next step, dephosphorylation occurs. You can understand that the moment the dephosphorylation occurs, an enzyme phosphatase is involved. So there is this dephosphorylation and CDK1 is totally inactivated. During this process, the cell has traveled through G1, S and is gradually moving towards G2. As the cell enters G2, cyclin B meanwhile is being synthesized, and just at the outset of mitosis, cyclin B binds with its partner CDK1. So we have this MPF dimer. The cell has not entered mitosis yet. The MPF dimer is ready, but the dimer is not active. For the activation of the dimer, you can see that there is an enzyme mentioned here, V1. So this enzyme is coming into play. This is a protein kinase because it will add phosphate to those 3 amino acids I mentioned at the start of this cycle. So this V1 protein kinase, what it will do, it will add phosphate to these 3 amino acids. It is adding phosphate to threonine 14. It is adding phosphate to tyrosine 15. And it is adding phosphate to threonine 161. So we have a dimer where the 3 amino acids of CDK has been phosphorylated, but yet this dimer is not active. The thing is that when this phosphorylation is occurring, there are 2 categories of phosphorylation occurring. This is known as the activating phosphorylation. That means when you're adding phosphate to threonine 161, the enzyme is supposed to be activated. But simultaneously adding phosphate to these amino acids, that is threonine 14 and tyrosine 15. When we are adding phosphates to these 2 amino acids, this phosphorylation is known as inhibitory phosphorylation. So understand the fact that V1, it is a protein kinase and it is adding 3 phosphates. One is the activating phosphate and the other 2 is the inhibiting phosphate. Since this inhibiting phosphate has been added to CDK1, even though there is this activating phosphate in threonine 161, the enzyme cannot be activated because there is an inhibition occurring from these two phosphorylations. So for the enzyme to be perfectly activated, those two phosphates needs to be removed. And thus comes a phosphatase which is known as CDC25. So the CDC25 is a phosphatase. By the name you can understand, it will remove the inhibitory phosphorylations. So what happens? In the next step, the CDC25, as you can see, if you look into this picture, it has removed those inhibitory phosphates from CDK1. The moment those inhibitory phosphates have been removed from CDK1, mitosis starts, MPF is active. So this is synonymous. MPF is active and mitosis starts, cell enters mitosis. So we understood two vital points, that during the process of MPF activation, there is one which is known as the activating phosphorylation, and there is one phenomena which is known as the inhibitory phosphorylation. This particular picture shows that in very clear manner. You see that mitotic cycline binds with CDK1. The moment mitotic cycline binds with CDK1, we do not have an immediately active MPF. What happens? There is a kinase which induces phosphorylation. So we have three phosphorylations. In this picture, the inhibitory phosphorylation has been shown as one. So we have an activating phosphorylation and we have an inhibiting phosphorylation. In this condition also, as you can see, the mitotic Cdk is inactive, even though it has paired with its cycline partner. In the next step, what we are seeing, that the CDC25 removes this phosphate, the inhibitory phosphates, and the moment these inhibitory phosphates are removed, as you can see in the picture, we are getting active mitotic cycline CDK partner. So this is one important point. So we have a activating phosphorylation in case of CDK1, and we have a inhibitory phosphorylation in case of CDK1. When there is this kinase enzyme, it adds phosphate to both the inhibitory site and the activating site. When we have the phosphatase enzyme active, it removes, as you can see by the arrow, it is removing the inhibitory phosphorylation. So right side, they have shown this picture. You can see the activating phosphorylation threonine 161 and the inhibitory phosphorylation. We can see that phosphorylation of the three sites are occurring by V1. Actually, there is another enzyme involved in this phosphorylation, but let's concentrate on what we have studied. So the phosphorylations are being taking place. All the three sites are being phosphorylated, both activating and inhibitory. The MPF is not active. Only when the CDC25 removes the inhibitory phosphate, we get an active MPF. Now what happens? The moment we get an active maturation promoting factor, you can see that the mitotic protein kinases like CDK1, Aurora kinase is another mitotic protein kinase, and polo kinase is another mitotic protein kinase. So remember that whenever the MPF is entering mitosis, as you can understand by the diagram, that the MPF molecule is active. That means that cyclin B is now regulating the activity of the cyclin dependent kinase. And as the name of this enzyme suggests that these are kinases, so just they will go on adding phosphates to their substrate. Aurora kinase and polo kinase are two more kinases that are activated during this process. And you can see what does this MPF does. Chromatin condensation. If you remember your stages of mitosis, so the first that occurs is chromatin condensation, breakdown of nuclear envelope, fragmentation of Golgi apparatus, and spindle formation. So during chromatin condensation, as you can see, it is phosphorylating the substrates such as condensins and cohesins. So the chromatin is condensing, and the nuclear envelope breakdown, phosphorylation of the lamines, the nuclear pore complexes, and the inner nuclear membrane proteins. Fragmentation of Golgi apparatus, for that it is for phosphorylating the Golgi matrix proteins. And finally, we are having spindle formation. So phosphorylation of centrosome, kinetochore, and microtubule associated proteins. So the moment MPF is activated, it brings about all the phosphorylation of all those proteins that gradually will start the different stages of mitosis. So if we now summarize the cyclines and the CDKs we have studied. Today we studied the M-phase cycline CDK complex, that is cycline B CDK1. Earlier we had studied the G1S cycline CDK complex, that was cycline D CDK4/6. And in the S phase, if you remember, we studied another cycline CDK complex, that is the cycline E CDK2. Remember that each cycline has a specific role. Each cycline is operative in a specific stage. For example, the mitotic cyclines are operative in G2M transition. The moment the cell moves out of M phase, the cycline B part is degraded. The G1 cyclines, that is the cycline D CDK4/6, is operative in the transition of the restriction point. Once the restriction point has been traveled by the cell, then automatically this cycline D CDK4/6 becomes non-functional. So another point that is to be also remembered, that the cycline CDK complexes are partners. The CDK part is the enzyme part, the cycline part is a regulatory part. And the cycline part, the life of the cycline part is very small. They are present during a particular stage, and the moment the cell moves away from that stage, they are automatically degraded. CDKs are enzymes, they're comparatively stable, but alone they are non-functional. Alone without their partner cycline, they are inactivated. Now this slide shows you that the different regulation of the CDK activity at multiple levels, how the enzymes are regulated. The first one, the first picture, as you can see, is what we studied today, is the cycline B CDK1 complex. We understood that there are a set of enzymes, the kinase and the phosphatase enzyme, that controls the cycline B CDK complex. The kinase enzyme phosphorylates both the activating and the inhibitory sites. The phosphatase enzyme removes the phosphorylation from the inhibitory sites, bringing about the activation of the cycline B CDK1 complex. So one set of control over the cycline CDK complex is done by the enzymes kinase phosphatase itself, which we studied today. Then there is another set of control that is often observed in case of cell cycle is proteolysis. Today I said that the moment MPF enters, as a result of functional MPF, the cell enters mitosis, and the moment the cell passes through the stage of anaphase, the cycline B is instructed to be degraded, which means that since the cycline B will be degraded, the cell will automatically have to move out of mitosis. Only again when the cell will move from mitosis G1S to G2, then only cycline B, by that time fresh cycline B will be synthesized, and that will bind to CDK1, and then again we'll get this active MPF. So proteolysis is a second form of control that is done by the cell for the passage of the cell from G1S, G2 to M in a defined manner. Third kind of control is inhibitor binding. If you remember, while talking about cycline E CDK2, I had talked about the inhibitors present that binds to active cycline E CDK2 complexes in the S, G2 or M phases, that is P27. These inhibitors can only be removed from these complexes when these complexes reach that stage where they need to be activated. For example, we studied the inhibitor P27 for CDK2 cyclin E complexes, and P27 was only removed only when the cell is on the verge of entering the S phase. So inhibitor binding is another important point by which these different partners of cyclins and CDK are controlled. And finally, another control that is done on these cyclin CDK complexes are the localization. Where is this cyclin present? So remember that in G2, most of the cyclin B that has been synthesized is present in the cytoplasm. The cell is on the verge of entering mitosis. It is during this stage that the cyclin B moves from cytoplasm into the nucleus, where it will bind with the cyclin CDK to form the active MPF. So subcellular localization, where this cyclin is present. During the stages, for example, in cyclin B, G1, S, G2, it is present in the cytoplasm. And just at the start, at the outset of mitosis, cyclin B moves into the nucleus to bring about its action. So these are the four molecular mechanisms by which these enzyme partners, CDK cyclins of different stages, are controlled. And once more to revise, we have M-phase cyclin, CDK1 cyclin B, the G1 cyclin that helps in transition of the restriction point, cyclin D, CDK4/6. Again, the entry of S phase, cyclin E, CDK2. The transition from S to G2, cyclin A, CDK1, G2M transition, again, cyclin B, CDK1. MG1 transition is indicated by the fact that the moment cyclin D is degraded, the cell exits mitosis. So with this, we move on to the next part. So this is the engine. In this cellular engine, remember two areas which are important for your syllabus. One is the control of the MPF. It enters mitosis, activated, moment cyclin is degraded, inactivated MPF. And the other important control is the RB, retinoblastoma. As long as there is cyclin D, normal condition, as you can see, the cell at the outset of G1 RB is dephosphorylated. So it binds with E2F. The moment there is growth factor signaling, cyclin D increases, it phosphorylates RB, as a result of which E2F is free. E2F is capable of resulting in transcription of all those proteins required for the S phase. So these are two very vital controls that are present in our cellular engine. And another control is the control of DNA replication, where when we are having a very low CDK level, the loading of origins are taking place, and these loaded origins can only initiate replication at the outset of S phase. And this is also in nutshell that different cyclins are operative. So in most cases we have this, there are two important things that needs yet to be discussed. That is this anaphase promoting complex, which I'll not take up today. And there is another point that needs to be discussed is the DNA damage checkpoints. So we have done the basics of cell cycle. We studied how cell passes from G2 to M. We studied how cell passes from M to G1. We studied how the cell moves through the restriction point. We have studied how DNA replication is restricted to S phase only. Initiation of replication is restricted to S phase only. And now we need to study two other vital aspects of cell cycle, is the checkpoints. So I'll stop my class today. We'll start with the damage checkpoints next day.